Hello, beautiful people. Thank you and welcome back. Sorry for being a little bit away. I was sick. But now that I'm back, it's very timely because I get to talk about the new change in the status of peptides. Last week, an FDA committee voted to recommend 6 peptides for legal compounding, including 2 of the most popular ones, BPC-157 and TB-500, which sounds like tuberculosis 500. And, within hours, a headline collapsed into one word, which is approved. But this word approved is doing a lot of heavy lifting, and it is not what you think it might mean. I am a pharmacist and also a medical science liaison. I read a lot of clinical trials, and then I translate what was in the clinical trials into something that people can actually understand. So hopefully I can get you to understand what has happened and what are the evidence behind those peptides and why you should or should not be taking them and what this regulatory approval really means. And first thing before we dive into the history and the evidence is that having a study is not the end of the conversation. It does not mean the work is done, the evidence is out there. Usually having one study in a molecule is the beginning of a series of many things that we need to elucidate about this molecule. What dose, what clinical benefit, what route of administration, in what species does this work for, humans or rodents? And what kind of endpoint we're measuring for, and what are the controls that we're measuring against. And so, in light of that, having a study could mean a number of things, and just because something has had a clinical study, it really doesn't mean what you think it means. So, hold on to that thought as we dive deeper into the history and the evidence. First, I want to go back in time and take a look at the history of regulation of peptides because that is going to be important in talking about what exactly the change in regulation means today. Back in 2023, FDA put 19 peptides into something called Category 2. Category 2 essentially means we have safety concerns, don't compound these. And then in early 2026, the companies that nominated those peptides withdrew the nomination and FDA removed them from Category 2. Now, removing does not mean approval. Coming off of the list is not the same thing as being cleared for use. It just sort of moved them back into limbo. And then on July 23rd and 24th, 2026, the FDA's Pharmacy Compounding Advisory Committee, the PCAC, reviewed 7 peptides and voted to recommend 6 of them for the 503A bulk drug substance list. Those 6 were PCB-157, TB-500, KPV, MOTS-C, CMAX, and Epidolone. And one of them, DSIP, was voted down. What does 503A really mean? The 503 list is a roster of ingredients and compounds that compounding pharmacies are legally allowed to prepare. It's a decision about whether or not a pharmacy can compound the substance, not a decision about its use, its safety, nothing like that. And it's definitely not a determination of whether or not the substance is effective. To determine whether or not something is effective, it would have to go through FDA's drug approval process, which takes months, years, fully randomized controlled trials, and a demonstration of both efficacy and safety. So those are 3 separate things coming off of Category 2, being approved for 503A, and actually being FDA approved. And those 6 peptides are most only cleared the first 2 categories. They have not been FDA approved. And then also the vote was non-binding, which means that the FDA can accept or reject. We'll see in the coming weeks what the FDA decides to do. And even if the FDA agrees, it still has to open a formal notice and comment period for the public as well as other healthcare professionals to comment on. And that process usually takes months. So the earliest that pharmacies can compound would be like late 2026, if not pushing into 2027. And here is a curious part, which is that the advisory committee actually overruled FDA's own scientists and clinicians who had recommended against these peptides. So what you're looking at is not a scientific consensus that these peptides work. It is a regulatory access objection made over the agency's own reviewers. So that's the history and the context. Let's take a look at the evidence and why FDA's own reviewers might have said no to these peptides. We'll only look at 3 of them though. If you guys want, I can break down all 6, but in the interest of time, let's just do the 3 most popular ones. Peptide number 1, and undisputedly the most popular one out there on the internet, BPC-157. It's been called the Wolverine molecule. I mean, it would be pretty cool if you could actually turn yourself into Wolverine. I did a short video on that on my Instagram, but for the sake of YouTube long form, let's dive deeper into this. BPC-157 has been promoted to do anything from general inflammation to healing cystitis to tendon and ligament repair to better recovery after sports, anything and everything. Now, mechanistically, it is associated with angiogenesis, which is a formation of new blood vessels, along with nitric oxide upregulation and promotion of growth factors. And in mice, BPC-157 accelerated tendon growth, ligament healing, muscles, nerves, guts, anything and everything. So if you're a mouse, this is really good news. And as a human, when you're reading this, you might get really, really excited. Here's a question that I always ask, which is what happens in humans because we're not mice? Surprise. And here is where the literature and the evidence sort of falls apart because it might have had hundreds of rodent data, but in humans, only 3 trials. That's it. And they're all published by the same group of people, which is a little bit sus. You always have to think about those things because if this thing is so miraculous, generally we would see multinational, clinical trials, several universities across the world will be jumping on this. And those clinical trials were not well done. So let's go through them. There's one about knee pain, which involved 16 patients. One about interstitial cystitis in women, which only had 12 women. And a safety study of the use of IV BPC-157 in 2 adults. 2. First, if you add it up, there are fewer than 30 people across the entire world. And secondly, these are not randomized controlled trials. There is no placebo arm. And these are open label studies, and open label studies are not that great when it comes to evidence generation. Why might that be? Well, it's because of something called placebo effect. It means that sometimes when we really expect there to be benefit, we want it so badly that subconsciously our brain is primed to expect that something might happen. And secondly, just because something might seem like it works for one person does not mean that it will work for another person. And randomized controlled trials over a large population is really good at assessing and averaging out everyone's little individual differences. And thirdly, because you're going into this knowing that you took the drug, you may positively attribute any change to the drug as opposed to, you slept better the previous day, or had more exercise, or just random things, because our bodies are not static. We don't all wake up feeling the same every single day. And I could also go on about why the knee pain study was particularly not well done. It was pretty much a chart review. It wasn't a standardized study with a standard endpoint. It was just oh, hey, these people happen to take some BPC-157 and let's ask them 6 months to a year later, did that help you in terms of knee pain? In which time, in its magnificent glory, the passage of time could heal injuries, right? Don't get me started on that. And so, but you might look at it and be but there's so many rodent data. I mean, why can't we rely on rodent data? Well, rodent healing models are actually very poor predictors of how it will do in humans. Species metabolize things differently, or genetically different. The dose doesn't scale linearly. For example, anyone who has pets will know that the dose that your dog and your cat may need to treat their thyroid problem is a lot bigger than the dose that you and I might need for a thyroid problem. The injury models used in those rodent trials tend to be artificial and acute, which is very different from like a chronic thing in humans. And the history of medicine is a graveyard for molecules that looked really promising in rodents, but didn't do anything in humans. So, like, am I gonna stop you from taking it as long as it's compounded correctly? No, I'm not gonna stop you, but if you want to be your own human guinea pig, maybe think twice. So, what's a verdict on BPC-157? It is biologically interesting, but it lacks the large-scale human data. Now, peptide number 2, TB-500, which always sounds like tuberculosis. And it's usually stacked with BPC-157 as one of the healing molecules. Everyone who's promoting TB-500 talks about thymosin beta-4. Thymosin beta-4 is an actual protein that is present in almost every cell in your body, and it is involved in things like actin formation, regulation, cell migration, wound healing, and it has a lot of research behind it. It is a real thing. But TB-500 is not the same thing as thymosin beta-4. It is a synthetic fragment of thymosin beta-4, and that fragment corresponds to the actin binding region. And this difference matters because almost all of the human clinical trials use the full thymosin beta-4 molecule as opposed to the small fragment of it. So the clinical evidence that people in the wellness industry cites for the evidence of TB-500 in humans was using a different molecule than TB-500. Now, I do want to make it clear that just because something is a shorter fragment of a larger molecule does not mean that it won't work. So we see that with heparin, which is an anticoagulant that was used very commonly, probably like 10, 20 years ago. But the problem with heparin is that, it's large molecule and you have to dose it very, very specifically. And it was a pain to dose. And then some companies came along and chopped it into smaller fragments called low molecular weight heparin. And now there are 3 different low molecular weight heparins on the market that are being used for anticoagulation. So first and foremost, I am not saying that just because something is a smaller fragment of a larger protein that it won't work. We have seen this in medicine. But what I am saying is that, as is the case with heparin, dosing was very different. Therefore, safety was very different. You could not dose low molecular weight heparins the same way as heparin. Heparin required dose titrations and calculation, and low molecular weight heparin it was a subcutaneous injection, either like once a day or twice a day, and it was weight-based as well. It's all very, very different depending on the weight of the molecule, the size of the molecule. And so different size means different pharmacokinetics, means different behavior, means different safety, and also different efficacy. Citing trials that had the big molecule to justify its use as a smaller fragment is just sort of bait and switch. But even if we say that the fragment behaves the same way as a parent molecule, parent protein, well, how well did thymosin beta-4 do in human studies? Not that well, actually. The most that thymosin beta-3 did was ophthalmic, like for your eyes, for your dry eyes. It's a formulation called RGN-259. It is an ophthalmic formulation used for dry eyes and for a corneal condition called neotropic keratopathy. And that formulation went into phase 3 trials. The ARISE-3 dry eye clinical trial missed its co-primary endpoints, which means that it did not meet the primary endpoint, which means it failed the trial. There was not sufficient evidence to see a clinical benefit with the use of this molecule. And then when they used it for corneal keratopathy, it also missed its primary endpoint. And the company attributed that to having a large placebo response. I'm telling you, placebo response is a very real thing, and sometimes just because people expect things to happen, they will try to justify any little change, especially something as like I don't want to say nebulous, but something hard to measure, like dry eyes. In another word, for both dry eyes and corneal keratopathy, this formulation did not work better than placebo. There was some interest for its use in things like wound healing, ulcers, and things like that, but as of now, it seems like the development for thymosin beta-4 has pretty much stalled. So, TB-500 is not the same thing as thymosin beta-4. And thymosin beta-4 failed 2 of its clinical trials, and it's sort of in a limbo. And 3, there are 0 completed trials in phase 2 or in phase 3 of TB-500 in humans for tendons, ligaments, muscle recovery, things like that. And for the TB-500 and BPC-157 combination that's been everywhere on the internet has never been tested as a combination in humans at all throughout all of the clinical trials that are out there. So there you go. Not only was the molecule not the one that's studied, the one that was studied failed its clinical trials. And finally, let's talk about the combination of CJC-1295 and ipamorelin. And this one's actually kind of interesting because it does have human data. It just didn't prove what it's being touted to do. So both of these molecules are growth hormone secretagogues. CJC-1295 is a synthetic version of growth hormone releasing hormone, the GHRH. Say that 10 times as fast. Growth hormone releasing hormone, GHRH. And usually the GHRH signals to the pituitary, somewhere there, to release growth hormones, growth factors. Ipamorelin works similarly, but it works on the ghrelin receptor, and it also triggers the release of growth hormones. So the logic of stacking both of them and using them together is that they will both provide growth hormones. Now, there is an actual double-blind, placebo-controlled clinical trial for CJC-1295. Subcutaneous CJC-1295 produced a sustained dose-dependent increase in growth hormone, roughly 2 to 10-fold, and in IGF-1, roughly 1.5 to 3-fold, lasting several days, and it was reasonably well tolerated at the studied dose. So case closed? Turns out not so much, because the trial measured a surrogate endpoint. It proved that CJC-1295 raised growth hormones and IGF-1 in your blood, but it did not prove that just by raising these would make you healthier, stronger, leaner, or that it's safe to do. A surrogate marker is a lab value that we associate with a real outcome, and we hope that their correlation is positive. But correlation does not mean causation, and just because a surrogate marker does something doesn't necessarily mean that you'll see an actual outcome. I mean, the world of medicine is also full of molecules and drugs that changed the surrogate marker but did nothing for the outcome. Like the world of cardiology is full of it as well. So what does that really mean? Well, it means that the controlled human studies for which this combination is being promoted for, so like being leaner, stronger, faster, etc., doesn't actually exist. Also, the combination of CJC-1925 and ipamorelin has not been studied in humans. The stacking of these two molecules is theoretical and hasn't been proven. And when it comes to safety, here is where nature paints us a negative story, because we do have a known condition in which there is excess secretion of growth hormones. It's called acromegaly. And sustained supraphysiological growth hormone drives insulin resistance, which means diabetes. And IGF-1 is a growth factor, which raises long-standing theoretical concerns about promoting the growth of things you don't want promoted. The proposed safety advantage of secretagogues that IGF-1 feeds back onto itself to cap growth factor release and giving it a physiological ceiling is a theoretical one as well. It's a theory. It's reasonable theory, but it's still a theory, and it hasn't been demonstrated in long-term human study. So what does that mean when it comes to peptide stack number 3? It's like, there are actual human studies on this, but the safety is really worrying. And also, we still don't have human trials on the thing that they're promoting it for. And here's the thing about FDA approval, right? When I was working as an MSL, it is so strict what you are allowed to promote drugs for. All of these things that I'm talking about would have been considered off-label, and drug companies would have been given a heavy fine and a slap on the wrist, and probably been told to never do this again, etc. It's drug companies, if they were to promote off-label use the same way that these wellness industries and peptides people have been doing, would have been given a very, very, very heavy fine. Because when you're going through FDA approval, and it's the same thing across the world, every country has its own health regulators, right? And these health regulators look at the clinical evidence, look at the endpoint, because the endpoint is what we're trying to prove that this drug is good for. And so these endpoints are usually hard outcomes. So in the case of cardiology, it would be major adverse cardiac events, what we call MACE. So they're stroke, heart attacks, atherosclerotic events. And when a drug proves to be effective in lowering the MACE, then the regulatory agencies can say, yes, this drug is effective in reducing the exact outcome measures. They're not allowed to say, oh, look at this secondary endpoint of also having better blood pressure. Well, it's a secondary endpoint. It's not in the primary endpoint. Therefore, the study is not powered to assess the secondary endpoint. Therefore, you cannot prove that this drug is also good for lowering blood pressure. And this submission process is so strict. And I know that the stereotype is that, drug companies have no scruples, or that sales reps and things like that would just say anything. That is not the case. There are many regulations about what drug reps and MSLs are allowed to say and not allowed to say. So drug reps can only say what is on the product monograph. That means essentially what's in the clinical trials. Medical science liaisons are allowed to say more about other data, about off-label data and uses, but only if the physician requests it. If we're going around spewing things about how this drug is amazing for all of those things because here's this one little anecdotal evidence from Korea, the company would be shut down. We get fired. And yet these wellness influencers are just allowed to say anything and everything. Anyhow, rant over. So what you need to keep in mind every time you encounter these wellness gurus touting the next thing is, has it been tested in humans or just rodents? What's the actual endpoint that it was tested against? What was the actual molecule? What was the dose? What was the route of administration? Was the actual molecule being used or not? Otherwise, you're sort of being a lab rat for yourself, which, is fine. But another thing I want to say is that most of the time, after the FDA approves a molecule and it is now on the market, there is a push to get safety data. So even if an effect is expected, if you take the dose the wrong way or things like that, it has to be reported to the company's safety side of things so we can monitor and we can, assess safety and things like that. But that is not the same thing as when you're taking something from a compounding pharmacy. There is no one, no company that is responsible for any ill effect that might come. Again, hopping off my soapbox. If I'm on my soapbox, I feel like we'd just be here forever. Let me be clear about where I actually am because I'm not here to say no to peptides. I'm not here to fearmonger. I'm not here to, tell you what you should and shouldn't do. If you want to inject it into yourself, as long as you're like using a version of it that's compounded correctly and doesn't have like heavy metals in it, great, you do you. But do be aware of the safety. And I'm not saying all peptides are bad because evidently the GLP-1 agonists are peptides and they have been working wonders. But they have had a lot of clinical trials behind them before they were even approved to be on the market. And that's the point. The problem was never about the molecule in itself, it was about the data, the evidence to hype ratio, and the gray market and dubious safety. Of course, the recent FDA change may prove that over time safety will get better. It may improve product quality, and it likely will improve quality if compounding pharmacies are making it instead of, I don't know, buying it from China. But if you're considering getting into peptides, you should have a conversation with your clinician and you should think about the long term safety data or the lack thereof. Anyhow, if you've made it this far, thank you so much for watching. I hope I didn't bore you with my little rants here and there. And let me know in the comments what molecule you think I should do next. And I'll see you next time. Thank you.